Hey guys, welcome to another episode of the Human AI podcast. Today we're going to talk about liver and metabolism. I'm here with Grok, an AI. If you're unfamiliar with how the structure of the podcast goes, AI and I talk. AI explains these topics to me. I'm very unfamiliar with these topics and I can hopefully gain some insights. Please take everything that the AI says with a pinch of salt. It can be wrong at times. However, if you cross-reference things and you do proper science in your own time, then this might be a valuable addition for you. Okay, well, having said that, Grok, let's start with today's topic. So, why is metabolism important, Grok?
Metabolism is crucial because it's the set of chemical processes in your body that keeps you alive, converting food into energy for everything from breathing to moving to thinking. It's like your body's engine, regulating how efficiently you use nutrients, maintain organ function, and manage waste.
And why do we need energy?
Energy is essential because it powers every function in your body, from basic stuff like keeping your heart beating and cells repairing, to more active things like walking, thinking, or even digesting food. Without it, your organs couldn't operate and you wouldn't be able to grow, heal, or respond to your environment.
Okay, and let's talk about ATP then. We always hear about ATP when it comes to energy. What's ATP? What's the structure of ATP? How does it function?
ATP, or adenosine triphosphate, is like the body's energy currency, a molecule that stores and releases energy for nearly every process in your cells. It's made of an adenine base, a ribose sugar, and 3 phosphate groups linked in a chain. When your body needs energy, ATP breaks one of those phosphate bonds, turning into ADP and releasing energy to power things like muscle movement or nerve signals. Your cells constantly recycle ATP, recharging it using energy from food through processes like cellular respiration in the mitochondria.
How does magnesium fit into the picture of ATP?
Magnesium plays a key role in ATP function because it stabilizes the molecule's structure and enables its energy transfer. It binds to the phosphate groups in ATP, forming a magnesium ATP complex that makes it easier for enzymes to break the phosphate bond and release energy for cellular processes.
I don't understand. Tell me a bit more about magnesium. So it's ATPs as a complex with magnesium, and the magnesium interacts with the oxygens of the triphosphate chain. It makes it susceptible to cleavage. What's going on?
You're right to zero in on that. Magnesium interacts with ATP by binding to the negatively charged oxygen atoms and the phosphate groups of the triphosphate chain. This binding does a couple of things. It neutralizes the negative charge, making the ATP molecule more stable, and it positions the phosphate bonds in a way that enzymes can more easily access them. This setup makes it easier for the enzyme to cleave the bond between the second and third phosphate, releasing energy and turning ATP into ADP. Without magnesium, the bond wouldn't break as efficiently, and your cells would struggle to access the energy ATP provides. It's like magnesium is the key that unlocks ATP's energy potential.
I see. Okay, now let's talk a bit about metabolism, anabolism, catabolism. Explain us the differences.
Metabolism is the whole set of chemical reactions in your body that keep you alive, and it's split into 2 main processes, anabolism and catabolism. Anabolism is about building up, using energy to create complex molecules like proteins or fats from simpler ones, like when your body makes muscle tissue. Catabolism is the opposite, breaking down complex molecules, like food, into simpler ones, releasing energy through its cells. Think of digesting carbs into glucose for fuel. Anabolism stores energy for growth and repair, while catabolism provides the energy to power your body's functions.
Okay. Let's talk now a bit about mitochondria and about the structure of mitochondria and about their membrane.
Mitochondria are your cell's powerhouses, where most ATP is made. They're small, bean-shaped organelles with a double membrane, an outer membrane that's smooth and permeable, letting small molecules pass, and an inner membrane that's highly folded into structures called cristae, which increase surface area for energy production. The inner membrane is packed with proteins and enzymes crucial for the electron transport chain, where ATP is generated through a process called oxidative phosphorylation. Between the membranes is the intermembrane space, and inside the inner membrane is the matrix, where the citric acid cycle happens, breaking down nutrients to fuel ATP production.
Okay, interesting. And why do we want it to be selectively permeable?
So, we were just diving into the structure of mitochondria and their membranes. The selective permeability of the mitochondrial membranes, especially the inner one, is key because it tightly controls what goes in and out to keep ATP production efficient. The outer membrane is more permeable, letting small molecules like ions pass, but the inner membrane is super selective.
Yeah, but why do we want it to be selective to have an efficient ATP production or whatever?
The inner mitochondrial membrane selective permeability is crucial because it creates a controlled environment for ATP production. By tightly regulating what enters and exits, like protons and ADP, it maintains a proton gradient across the membrane, which drives the proton motive force. This force powers ATP synthase, an enzyme that churns out ATP during oxidative phosphorylation. If the membrane were too leaky, the gradient would collapse and ATP production would stall, like a dam failing to generate power because water flows too freely.
Okay, interesting. Let's talk a bit about cytosol and mitochondria. What part of the metabolic process happens in the cytosol and what part happens in the mitochondria?
In the cytosol, the first stage of metabolism, called glycolysis, happens, where glucose is broken down into pyruvate, producing a small amount of ATP and NADH without needing oxygen. The mitochondria take over for the next stages. Pyruvate moves into the mitochondrial matrix for the citric acid cycle, which generates more NADH and FADH2, and then the electron transport chain on the inner membrane uses those to produce a ton of ATP through oxidative phosphorylation. So, cytosol handles the initial breakdown, while mitochondria crank out the bulk of the energy.
So, cytosol, we have glycolysis, and what do we have in the mitochondria?
In the mitochondria, you've got two main metabolic stages. The citric acid cycle, also called the Krebs cycle, in the matrix, which breaks down pyruvate to produce NADH and FADH2, and the electron transport chain on the inner membrane, which uses those molecules to drive oxidative phosphorylation, pumping out most of the cell's ATP.
So TCA cycle and ETC. Okay. And why do we have it this way and not the other way around?
The TCA cycle, or Krebs cycle, happens in the mitochondrial matrix because it needs specific enzymes and conditions to break down pyruvate into energy-carrying molecules like NADH and FADH2. The electron transport chain, ETC, follows on the inner mitochondrial membrane because it uses those molecules to create a proton gradient across the membrane, which drives ATP synthase to produce ATP efficiently. This setup maximizes energy output. The TCA cycle generates high-energy electron carriers, and the ETC harnesses them in a controlled way to pump protons and power ATP production. If it were reversed, with the ETC in the cytosol and TCA in the mitochondria, you'd lose the tight membrane-based proton gradient needed for ATP synthase to work effectively, and the process would be far less efficient. Plus, the cytosol lacks the specialized enzymes and structure that mitochondria provide for these reactions.
I still don't understand your answer, to be honest. I don't understand the answer.
All right, let's break it down simpler. The reason glycolysis happens in the cytosol and the TCA cycle and electron transport chain, ETC, happen in the mitochondria is because each process needs specific tools and conditions. The cytosol is like an open workspace where glucose can quickly get broken down into pyruvate during glycolysis, producing a bit of ATP without needing fancy equipment. Mitochondria, though, are like a high-tech factory. The TCA cycle needs the mitochondrial matrix because it's packed with enzymes that turn pyruvate into energy-rich molecules like NADH. The ETC needs the inner mitochondrial membrane because it's got proteins embedded in it that use NADH to pump protons and create a gradient, which is like a battery powering ATP synthase to make tons of ATP. If you tried to do the TCA cycle or ETC in the cytosol, it'd be like trying to run a factory without walls or machines. Too chaotic and inefficient.
Okay.
The
All right. Let's talk about cofactors now. So we have metal ions, we have coenzymes. Why do we differentiate between these two? And then why do we differentiate even more between co-substrates and prosthetic groups? Talk to me about what these are. So why are cofactors non-protein molecules necessary for enzyme activity? And how come vitamins, or how come most coenzymes are derived from vitamins? And explain this a bit.
All right, let's dive into cofactors. Cofactors are non-protein molecules that help enzymes do their job because enzymes alone often can't catalyze reactions efficiently without a little boost. These cofactors can be metal ions like magnesium or zinc, or organic molecules called coenzymes like NAD plus or FAD. We differentiate between metal ions and coenzymes because they work differently. Metal ions like magnesium and ATP stabilize charges or help with enzyme structure, while coenzymes often carry electrons, atoms, or groups during reactions. Now, coenzymes split into co-substrates and prosthetic groups. Co-substrates, like NAD+, temporarily bind to an enzyme, get modified during the reaction, and then leave to be recycled, like a shuttle carrying cargo. Prosthetic groups, like heme and hemoglobin, are tightly bound, staying put as part of the enzyme structure, helping repeatedly without detaching. Why are cofactors necessary? Enzymes are proteins, and their amino acid structures can only do so much. Cofactors add chemical tricks like electron transfer or stabilizing reactive intermediates that proteins alone can't handle. Without them, many reactions, like breaking down glucose or synthesizing DNA, would grind to a halt. As for vitamins, most coenzymes come from them because vitamins are dietary molecules our bodies can't make, and they're often converted into coenzymes. For example, vitamin B3 becomes NAD+, and vitamin B2 becomes FAD. These vitamins provide the chemical backbone for coenzymes to carry out their roles in metabolism.
But why the non-protein part? I don't get the non-protein.
The non-protein part of cofactors is key because proteins made of amino acids have limited chemical abilities on their own. Amino acids can form shapes and bind molecules, but they often lack the specific chemical properties like carrying electrons, transferring groups, or stabilizing high-energy states needed for certain reactions. Non-protein cofactors, like metal ions or coenzymes, bring those unique chemical capabilities. For example, a metal ion like zinc can act as a Lewis acid to stabilize negative charges, and coenzymes like NAD+ can shuttle electrons in ways protein side chains just can't. It's like enzymes are the tools, but cofactors are the specialized bits that make the tools work for tough jobs in metabolism.
I see. Yes. Okay. And now, talk to me about redox coenzymes. So, we have this electron transfer that you've mentioned, and then give us some examples like NAD+, FAD. What do these stand for? Because we're going to talk about them quite a bit. Yeah. And how come the electrons are transferred? Like in NAD plus and FAD, we have 2 electrons transferred, but the number of protons varies. NADH versus FADH2.
All right, let's get into redox coenzymes. These are molecules that carry electrons during metabolic reactions, especially in processes like energy production. They're critical for redox reactions, where one molecule loses electrons, oxidation, and another gains them, reduction. NAD plus and FAD are prime examples. NAD plus stands for nicotinamide adenine dinucleotide, and FAD stands for flavin adenine dinucleotide. Both are derived from B vitamins, NAD plus from vitamin B3 and FAD from vitamin B2. Here's how they work. NAD plus and FAD act like electron shuttles. In reactions like glycolysis or the citric acid cycle, they pick up electrons from molecules being broken down, like glucose. NAD plus typically grabs 2 electrons and 1 proton, forming NADH, with an extra proton, H plus, released into the environment. FAD, on the other hand, picks up 2 electrons and 2 protons, forming FADH2. The difference in proton pickup comes down to their chemical structures. NAD plus has a nicotinamide ring that binds a hydride ion, 2 electrons, 1 proton, while FAD's flavin ring can handle 2 protons and 2 electrons, making FADH2 neutral. This electron transfer is crucial because it feeds high-energy electrons into the electron transport chain in mitochondria, where they drive proton pumping to power ATP production. The varying proton numbers affect how much energy each coenzyme delivers to the chain. NADH generally contributes to more ATP than FADH2 because of where it feeds into the chain. It's like NADH and FAD are different types of fuel pumps, each delivering energy in a slightly different way to keep the cell's engine running.
Okay.
So we were just discussing.
Okay. And what is oxidative phosphorylation? What does it have to do with ATP synthesis? How is it connected with the reoxidation and redox of this NAD, FAD?
Oxidative phosphorylation is the process in mitochondria where ATP is made using energy from the electron transport chain, ETC. Here's how it ties in. NADH and FADH2, those redox coenzymes we talked about, carry high-energy electrons from reactions like the citric acid cycle. They donate these electrons to the ETC, a series of protein complexes on the inner mitochondrial membrane. As electrons move through the chain, they release energy, which is used to pump protons, H+, across the membrane, creating a proton gradient. This gradient powers ATP synthase, a protein that acts like a turbine, letting protons flow back into the mitochondrial matrix and using that energy to convert ADP into ATP. The reoxidation of NADH and FADH2, turning them back into NAD+ and FADH, happens when they lose their electrons to the ETC. This recycling is crucial because it frees up NAD+ and FAD to keep picking up electrons in metabolism, keeping the whole energy production cycle going. Without oxidative phosphorylation, your cells wouldn't get the bulk of their ATP, and metabolism would stall. It's like the final step in turning fuel into usable energy for your body.
Okay.
So we
Can
were
you
just
explain
talking.
this one more time, to be honest? Sorry, I lost focus for a second. Explain it, and I think you did a good job. Just tell it again.
No worries, let's go over it again. Oxidative phosphorylation is how your mitochondria make most of the ATP, the energy currency for your cells. It happens in 2 main steps on the inner mitochondrial membrane. First, NADH and FADH2, those redox coenzymes from earlier, carry high high-energy electrons from food breakdown and processes like the citric acid cycle. They hand off these electrons to the electron transport chain, ETC, a lineup of proteins in the membrane. As the electrons move through the ETC, their energy is used to pump protons, H+, out of the mitochondrial matrix, creating a proton gradient, like a dam holding back water. Second, this gradient drives ATP synthase, a protein that lets protons flow back into the matrix, using that flow to power the conversion of ADP into ATP. The reoxidation of NADH and FADH2 happens when they lose their electrons to the ETC, turning back into NAD+ and FAD so they can be reused in metabolism. It's like a power plant. NADH and FADH2 supply the fuel, the ETC builds the charge, and ATP synthase churns out the energy your cells need.
Okay, let's talk a bit about So 2.5 molecules of ATP are synthesized for each NADH reoxidase. It's so specific, and I wonder why. And 1.5 for FADH reoxidation.
Yeah, that 2.5 ATP per NADH and 1.5 ATP per FADH2 is super specific, and it comes down to how the electron transport chain, ETC, works. When NADH donates its electrons to the ETC, it feeds them into complex 1, which pumps more protons across the mitochondrial membrane than complex 2, where FADH2 donates its electrons. More protons pumped means a stronger gradient, which drives ATP synthase to produce more ATP, roughly 2.5 ATP per NADH because of the extra proton pumping. FADH2, starting at complex 2, pumps fewer protons, so it yields about 1.5 ATP. These numbers are averages, not exact, because the proton to ATP ratio depends on the efficiency of ATP synthase and the membrane's leakiness. It's like NADH is a high-octane fuel that gets more mileage, while FADH2
But
is...
I thought that you said FADH2 has 2 protons, which I kind of understood means more ATP. It obviously seems I fucked up in my understanding.
No worries, let's clear that up. The confusion likely comes from the protons FADH2 carries versus the protons pumped in the electron transport chain, ETC. FADH2 does carry 2 protons and 2 electrons when it's formed, but that's just its chemical structure from the citric acid cycle. The key to ATP productions isn't those protons, it's how many protons get pumped across the mitochondrial membrane by the ETC when FADH2 or NADH donates electrons. When NADH gives its electrons to complex 1 in the ETC, it triggers the pumping of about 10 protons across the membrane. FADH2, however, feeds electrons into complex 2, which skips complex 1 and pumps fewer protons, about 6. More protons pumped means a stronger gradient for ATP synthase to use, so NADH drives about 2.5 ATP per molecule, while FADH2 only gets about 1.5 ATP.
Okay. That's good. Okay, let's go to the big boss, or at least one of the big bosses, and analyze it a bit more. Glycolysis. Let's talk about the stages. So glycolysis is in the cytoplasm. I still don't understand why cytoplasm and not mitochondria. And it starts from glucose. Okay. Then we've got a few stages. Hexokinase will create glucose 6-phosphate. Okay. And ATP will be converted to ADP. Explain us up to this point what's going on.
Glycolysis happens in the cytoplasm because it's a quick, oxygen-independent process that doesn't need the specialized machinery of mitochondria. Think of it as a lightweight setup for breaking down glucose fast. Mitochondria are built for later stages, like the citric acid cycle and electron transport chain, which need their membranes and enzymes to maximize ATP production. The cytoplasm is just more accessible for glucose to start getting processed. Now, let's zoom in on the start of glycolysis. It begins with glucose, 6-carbon sugar entering the cell. The first key step involves hexokinase, an enzyme that grabs glucose and uses one ATP molecule to stick a phosphate group onto it, forming glucose 6-phosphate. This step does a couple of things. It traps glucose inside the cell by making it less likely to diffuse out, and it primes the molecule for further breakdown. The ATP is converted to ADP because it donates its phosphate group, acting like an energy investment to kickstart the process. It's like paying an entry fee to get the glycolysis party going, setting up glucose for the next steps, where it'll eventually split and generate energy.
Okay, and then we've got isomerase turning glucose 6-phosphate to fructose 6-phosphate. I wonder why isomerase and hexokinase are the right enzymes for these steps and how their names relate to their structure and function. Okay, after that, we get, again, conversion of ATP to ADP. And I wonder why we need to use ATP at this stage. And now we've got something called the committed step. Explain us why. And we've got PFK1. What is that? Phosphofructokinase. And then we have fructose 6-phosphate becoming fructose 1,6-biphosphate. So you have to explain to us these steps because they're very arbitrary.
All right, let's unpack this part of glycolysis step by step. After glucose 6-phosphate is formed, the enzyme phosphoglucose isomerase comes in and rearranges it into fructose 6-phosphate. Why this enzyme? Its name, isomerase, reflects its job. It isomerizes, meaning it reshapes the molecule without adding or removing atoms, just rearranging the structure. Glucose 6-phosphate has a 6-membered ring, but fructose 6-phosphate has a 5-membered ring, which is easier for later enzymes to work with. The phosphoglucose part of the name points to its substrate, glucose 6-phosphate, and its specificity in handling sugar molecules. Hexokinase, in the first step, is named for its role, hexo for 6-carbon sugars like glucose, and kinase because it transfers a phosphate group from ATP. Its structure is designed to bind both glucose and ATP, positioning them perfectly for the phosphate transfer. Next, fructose 6-phosphate is converted to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1, or PFK1. This step uses another ATP, which becomes ADP, to add a second phosphate group to the sugar. Why use ATP again? It's like an energy investment to make the molecule more reactive and unstable, setting it up to split easily in the next step. PFK1 is named for its role. Phospho for phosphate, fructo for fructose, and kinase for phosphate transfer. Its structure has a specific active site that binds fructose 6-phosphate and ATP, and it's tightly regulated because this is the committed step. Why committed? Once fructose 1,6-bisphosphate is formed, the molecule is locked into glycolysis. It can't easily be used for other pathways.
And why are the previous reactions, the ATP to ADP called priming reactions?
The earlier reactions in glycolysis, where ATP is converted to ADP specifically, the hexokinase step, forming glucose 6-phosphate, and the PFK1 step, forming fructose 1,6-bisphosphate, are called priming reactions because they prepare glucose for breakdown. By adding phosphate groups, these steps use ATP to make the sugar molecule more reactive and unstable, like winding up a spring. This energy investment traps glucose in the cell and sets it up to split easily into 2 3-carbon molecules later in glycolysis, which ultimately leads to energy production.
Okay, and tell me lastly about... Actually, guys, I've decided these videos are going to be 30 minutes long. Actually, no, let's finish this. Maybe it's going to be 31. Okay, to finish, just for the part 1 out of 2, fructose 1,6-biphosphate now will go to either DHAP or G3P. And we have aldolase. Why is aldolase the one?
All right, let's wrap up this part of glycolysis. After fructose 1,6-bisphosphate is formed, The enzyme aldolase steps in to split it into two 3-carbon molecules, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G30P. Why aldolase? Its name comes from its ability to catalyze an aldolase cleavage, which is a specific chemical reaction that breaks a carbon-carbon bond in a molecule like fructose 1,6-bisphosphate. The enzyme structure has an active site perfectly shaped to bind fructose 1,6-bisphosphate and cleave it symmetrically into DHAP and G30P. This split is crucial because these two molecules can then continue through glycolysis to produce energy. Aldolase is the right fit because its design ensures this high-energy molecule splits efficiently, setting up the payoff phase where ATP and NADH are generated.
Okay. Perfect. Well, guys, thank you for watching part 1 of this podcast. Make sure to watch part 2 soon.